Well, it's really great to be here. I'm glad everyone's participating. I'm excited to be one of the co-organizers and also have this opportunity to share with you some of my lab's own research that's relevant to this symposium. So today I'll just be sharing with you one vignette on the topic of human-specific alterations in brain cellular proportions. So one of the major goals of my lab is to try and understand what are the human-specific genetic and molecular features of the brain. If they exist, can we identify them, and can we use them to give us some insight into the behavioral features that set humans perhaps apart from some of our closely genetically related species, such as chimpanzee, as shown here. Now, as a card-carrying neuroscientist, I usually have to show one slide from Ramon Cajal, as indicated here in this slide. And Ramon Cajal and other neuroscientists now for over 100 years have been looking at the brain in one way. There are many ways to look at the brain, and that's with respect to trying to catalog and observe and give us information into the various cell types, not just in the neocortex, as indicated here in this slide, but throughout the entire brain. And in more recent modern times, current times, there have been a lot of efforts, cell atlasing efforts, using transcriptomics at the single cell level to build these atlases that define all of the various brain cells that are in the human brain, as well as in other species. And if we can do that in the human brain and in closely related species, then we might have some insight into cell types that might distinguish us from our closely related species, such as chimpanzees. And so this is just one effort through one of the atlasing efforts. And I think Ramon Cajal and his contemporaries would be surprised that by using transcriptomics, these atlasing efforts have identified thousands of cell types in the mammalian brain. Because for many years, we've thought of brain cell types in kind of a simplistic manner, that we have neurons and non-neurons. And over the years, various non-neuronal cell types have gone in and out of fashion. There's been some recent interest in microglia. Astrocytes tend to come and go within the neuroscience field, but I would like to make a pitch today for other cell types and subtypes of major cell types. And I'll be spending a lot of time in my talk focusing on oligodendrocytes, which, as shown in this cartoon, indicate that they are important for myelination of neurons, but I'd also like to show you or present evidence that they may be involved in other important aspects in brain function. And this is just another cartoon to indicate to you that, again, it's not just major cell types we care about, but subtypes of all of these major cell types as well. And so they're very varied, and we can look at them not just based on morphology, but also at the level of transcriptome, as these atlasing efforts have done. So like many probably in this symposium, my work has been inspired by the work of Mary Claire King and A.C. Wilson that was published now over 50 years ago, where they were looking at levels of proteins between humans and chimpanzees, found that they were very similar, and then came to the conclusion that their macromolecules are so alike that it must be regulatory mutations that account for their biological differences. And now we know, fast forward many decades later, after the sequencing of the human and chimpanzee genomes, that indeed at the level of the protein coding parts of genes, they're highly similar. But if we look outside of those parts of the genomes where there's less constraint, that's where we can see some variation. It's also how those genes are regulated, how they're turned on and off, how they're expressed, that may lead to the different phenotypes and perhaps functions and behaviors between species. And so for a number of years, we've decided now to make these comparisons across species at the gene expression, gene regulation level. And so this is just an example of looking at one part of the brain, in this case, the neocortex, the dorsolateral prefrontal cortex, among 3 species. Now, typically, we would want to have a minimum of 3 species, if not more, so we can assign changes in gene expression specifically to the human lineage. And so we would look for genes that are similar between chimpanzee and our outgroup, rhesus macaque, and then genes that are different between human and chimpanzee, and as well as human and macaque. And in this case, we can then assign that gene expression change to the human lineage. And we want to do this not just broadly, but try and really intersect this idea of many different types of cells in the brain with changes of gene expression. And a number of years ago, we weren't quite able to refine it down to that subtype level, but we could get broad cell type comparisons by just simply looking at neurons and all the cells in the oligodendrocyte lineage. And we could do this by taking tissue from humans, chimpanzee, rhesus macaque. This is all post-mortem tissue. Isolate the nuclei from this tissue, sort using antibodies for cells that were neurons or cells from the oligodendrocyte lineage, and then sequence the RNA to get gene expression levels. And if we look at just the first principal components of these analyses, either in the neurons or the oligodendrocyte lineage, this basically recapitulates phylogeny, which was good. However, what we really care about are changes in gene expression, changes specifically on the human lineage. That's our interest when we look between these two major cell types. And using the number of millions of years that separate these species from common ancestors, we could define genes changing specifically in neurons or genes changing specifically in oligodendrocytes on the human lineage. And the changes in neurons were not surprising, and we were very pleased with those. But the fact that we got many changing in oligodendrocytes and perhaps an even more accelerated change in oligodendrocytes than neurons was surprising to us, because this didn't follow what had been previously shown in the literature. And so we thought perhaps this was technical issues. And so what we did was we looked at the previously published studies, including work that came from my own research, and we intersected those with these newly identified human-specific neuronal genes and human-specific oligodendrocyte lineage genes. And everywhere you see red here indicates a significant overlap. And as we expected, we found a significant overlap with the previously published neuronal genes. However, our new data in the human oligodendrocytes did not overlap with the previous published data sets as we had remembered. So again, we were still left with this feeling that, oh, perhaps there's some sort of technical difficulty in our approach because this hadn't been seen before. So right around this time, the field generally of genomics was able to isolate truly individual cell types and quantify the transcriptome within individual cells using what's called single-cell genomics. There are a number of ways of doing this. One way is through a microfluidics approach using a droplet-based capture of cells or nuclei. And a number of groups had published the first beginnings of these atlasing efforts to identify cell type transcriptomes in human and mouse brain, for example. And we could take those kind of ground truth cell type transcriptomes and use that to do what's called a deconvolution of our somewhat sorted or even a bulk tissue approach. And when we did that, we found something very striking in that the majority of signal that was in the published studies was really derived from excitatory neurons. And we now know, many years later of doing this single cell approach over and over again, that neurons express more genes and they express them at higher levels. And so if one was to take a piece of brain tissue and just do gene expression on it, the majority of the signal would indeed be driven by neurons. And so what we could then do was take our sorted data and apply the same deconvolution approach, and we could see that the neuronal sorted nuclei were driven by gene expression from neurons, and the oligodendrocyte sorted nuclei were driven by signals from oligodendrocytes. And so the previously published studies and a lot of the literature was basically unable to detect this non-neuronal signature because the neuronal signature was so high, it was masking it. So it wasn't a technical issue on this new approach. It was more a technical issue on what we had been doing previously in the field. So this was where we were a number of years ago, and we wanted to intersect this approach of this comparison across species and really look, again, identify human-specific gene expression changes, but look at all of the cell types, not just neurons and only the oligodendrocyte lineage, although we're primed to think about this and keep in mind all these subtypes. And so we had a study moving forward where we had the same approach of comparing human, chimpanzee, using rhesus macaque as our outgroup. But in this case, instead of using frontal cortex, we were using posterior cingulate cortex. The reason we used this part of the brain was, A, it was available. It's very hard to get these tissues from non-human primates. And B, we could be certain we were comparing roughly the same region across all of the species. It hadn't been looked at before transcriptomically, to our knowledge, across these species. And so we then took the tissue, we isolated nuclei, and we used this new approach of single cell transcriptomics, not the sorting approach, but looking at every individual nuclei. And then in this case, we could identify many different subtypes of the oligodendrocyte lineage because we were already primed to be thinking about oligodendrocytes in human brain evolution. So OPC is oligodendrocyte precursor cell, COP, committed oligodendrocyte progenitor, newly formed oligodendrocytes, mature oligodendrocytes, etc. And one of the first, before we even started thinking about genes, one of the first things we wanted to look at, because we, again, had kept all the cells, we didn't enrich for one or the other, was whether there were changes in proportions among all of these cell types across all of the species. And what we found, very strikingly to our eye here, was that there was an increased relative proportion of human, indicated here in blue, of these immature oligodendrocytes, not just the OPCs, but also the COPs, relative to the mature oligodendrocytes. We then quantified this and applied statistics to it and found, indeed, a significant increase of immature oligodendrocytes and a significant decrease of mature oligodendrocytes on the human lineage. We found this not only at the level of RNA, but also at the level of chromatin accessibility, because in the same tissue, we applied another approach of single nucleus ATAC-seq, which looks at open and closed areas of chromatin. So this was very exciting to us. But one of the caveats to this approach is that you have to take the tissue and dissociate it to do this experiment. And so being cautious, I thought perhaps we may have inadvertently only in the human samples taken perhaps more white matter or areas that would have more oligodendrocytes than the other species. So a way to validate this result without having to dissociate tissue is to carry out and in situ hybridization at single cell level. So in this case, single molecular fluorescent in situ hybridization, SM-FISH. And we could use a probe for immature oligodendrocytes, PDGFRA, or mature oligodendrocytes, MOG. And we did this in independent, intact tissue from both humans and chimpanzees. And if we quantified this, we could recapitulate what we found in the single cell approach with an increased immature oligodendrocyte ratio in the humans compared to chimp, decreased mature oligodendrocyte ratio, humans compared to chimp. So this is a really great validation. We were very excited about this. And it struck me that, this seemed like unusual that this hadn't been observed previously, perhaps in the literature, maybe due to, lack of technology or markers to distinguish these cell types. But also perhaps there was just some vagary of the posterior cingulate cortex. Researchers hadn't looked at this closely. So I thought, well, what if we looked at other areas of the brain? And so there was a publicly available data set from the anterior cingulate cortex from Philip Kaitovich and colleagues, where they had human, chimpanzee, and rhesus macaque single cell data. And using that published data and reanalyzing it, we were able to find also in this brain region, increased immature oligodendrocytes, decreased mature oligodendrocyte ratio. Now, you may ask, why didn't these individuals report this change in this ratio? Well, this was because in the years since they had published this, in the years where we were looking at this, it became apparent to the field and work out of my own lab that when one dissociates the tissue, the neurons which express more genes, more RNAs, all the way out to the synapse, you kind of open up those RNAs into the fluids, it gets encapsulated, and you have this issue of ambient RNA. So we found ways to clean data sets. We cleaned their data set and found this change in ratio. We also, in this case, wanted to confirm, again, in intact tissue samples, this finding. And so we got independent human and chimpanzee tissue samples, performed the same SM fish experiment, quantified it, and again found this increased immature, decreased mature oligodendrocyte ratio in humans compared to chimpanzees. So we were very excited about this. But again, being ever cautious about new findings, I thought, well, perhaps this is just something about the anterior cingulate. We looked in the posterior, the cingulate cortex in general. We had looked in the anterior and the posterior. What about other parts of the brains? Were there other data sets that we could query to see whether this held up there? And so we took our sorted data that I started this presentation with from the dorsolateral prefrontal cortex. We didn't have it at cell type resolution, but we could distinguish using this deconvolution approach, this ratio. And again, using this data, we could see this increased immature to mature oligodendrocyte ratio in the dorsolateral prefrontal cortex. There was also a publicly available data set from the Allen Institute folks looking in the primary motor cortex. They had human and rhesus macaque and also human and marmoset. And again, in these data sets, cleaning it up for ambient RNA, we could again find this same change in oligodendrocyte ratio specifically in the human brain. So this was very exciting. Everywhere we looked in the neocortex, we could see this change in ratio specifically in humans. What about outside the neocortex? Well, at the time, there were two publicly available data sets, one in the caudate nucleus, one in the dentate gyrus. And in neither case did we see a change in this ratio in these non-neocortical regions. So to summarize, what we found was that humans have this altered OPC and oligodendrocyte ratio, specifically in cortical regions. And across all of these data sets, now we have 4 species. We looked in 4 neocortical regions: posterior, anterior cingulate, dorsolateral prefrontal cortex, primary motor cortex, and 2 non-neocortical areas. So yes, in the neocortical areas, no in the other regions. Now, as all of these atlasing efforts across the entire brain are moving forward, we'll see how this ratio does or does not hold up across other parts of the brain. Importantly, we're also hoping to look to see how this ratio may or may not dynamically change over development, and how that, if there is a dynamic change over development, where it happens in the human brain development compared to other species brain development. So one may ask, well, if this is a ground truth about human oligodendrocytes, this change in immature to mature ratio, specifically on the human lineage, why would the human brains have evolved this? What could it be good for? So taking this image from a review now over 15 years ago, hopefully you can appreciate that mature oligodendrocytes are important for myelination. But as you can see along this whole lineage, there are other purposes for immature oligodendrocytes. Some of these red squares are indicating neurotransmitter receptors or signaling. And so we know that these immature oligodendrocytes serve other functions for at least interacting with neurons, let alone other cell types, in addition to myelination. So we've developed this hypothesis that there could be, that evolution could have occurred to have this increased potential, perhaps experience-dependent generation of mature oligodendrocytes, that there's this pool of immature oligodendrocytes that's sitting here, this increased proportion, waiting for some signaling, either from neurons or other cells, telling it to either myelinate or to do other things. And for example, OPCs have also been shown recently to be involved in pruning synapses. And this next slide shows some of the other functions of oligodendrocyte precursor cells. They're involved in hypoxia, in injury. And importantly, one point I want to make is this idea of demyelination. With respect to thinking about how this might be important for brain evolution, how it may intersect with brain disease, there is one human demyelinating disorder, multiple sclerosis, that has not been observed in any other species. And this is something that could very likely, of course, be observed pathologically. And so up to the current time, we have not seen this in other species. So it could be something about this change in the OPC to mature oligodendrocyte ratio in the human brain that makes the human brain at risk for multiple sclerosis and intersecting perhaps with other cell type evolved functions, such as in the immune system. So one other really final point I'd like to make about these data is we're doing it all in postmortem tissues. We're making these important findings, developing these hypotheses. But then the question would be, how could we test the hypotheses? We can't really do that ethically or for a number of reasons in postmortem tissues. So we can use in vitro models, and you've heard about stem cell models or will be hearing about it in this symposium. And so in this case, we can take iPSC-derived cells from both humans and chimpanzees, and our lab has developed a fast protocol for differentiating IPCs into both immature and mature oligodendrocytes alone without the need for other cells. And if we quantify that and compare between the two species, we can again see this change in ratio, an increased immature as indicated by PDGFRA, and decreased mature as indicated here by O4 and olig-2 positive cells ratio in a dish. So this cell type model alone recapitulates what we were able to observe in the in vivo tissue. So now we can use this to test our hypothesis about whether this ratio is important for example, the experience-dependent plasticity of neurons. If we co-culture them with neurons and mix and match species, human OPCs with chimpanzee neurons or vice versa. And we can also test the contributions of individual genes to this alteration ratio between the species. So it's very exciting times. So I've given you some data and information about changes in the oligodendrocyte lineage in these species, in these brain regions, but what data are we still missing? Well, all of these data were, at least in the neocortex, were derived from the gray matter. And we know the majority of, or a lot of oligodendrocytes are found in white matter parts of the brain. And so we need to be doing the same sort of cell type transcriptomic profiling in those parts of the brain, in the white matter tracts. We also need to understand if there may be differences across different regions of the brains, just like we did in the neocortex. What about different areas of white matter? Are all oligodendrocytes and their precursors the same transcriptomically? How do the proportions and gene expressions of these oligodendrocytes change over development? As I already mentioned, that would be very exciting to see where and when this occurs, and does it change when we're comparing across species, if we can actually generate those data across closely related species, such as non-human primates. So to summarize some of the take-home messages, it's very important when you're looking across species and transcriptomically to treat all cell types equally. If you want to observe these changes in proportions, think about how proportional differences could occur across regions, across development, across species. And I showed you some validation experiments using SM-FISH and how that validated the dissociation of the microfluidic single cell approach. But there are new technologies in play now, spatial transcriptomics, where you can have the tissue intact and measure the transcriptome of every cell. And I think that will eventually really be the best way to look at how these changes are occurring at the cell type level across tissues and across species. And no matter what you're using to generate the data, validation in an independent way is great. In this case, I showed you SM-FISH, and now we can test the functional outcome, test our hypotheses in a system that's perturbable, such as the iPS-derived approach. So I'd just like to, here's a picture of my lab from almost 2 years ago now. Thank everyone in my lab that participated. I have a great set of collaborators, many of whom contributed tissue. For example, Chet Sherwood and Bill Hopkins for the non-human primates. Sujin Yee's been a great collaborator, my funders. And I'm looking forward to the Q&A session at the end of this. And thank you so much for attending this symposium.